Good afternoon. I'm Dr. Kara Wilder, a technical writer at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Advancing Standards for Clinical Diagnostics, presented by Mary Ellen DeMars. Dr. DeMars is the Senior Director of the Standards Resource Center at ATCC. She has a PhD in virology and has over 25 years of experience in the development of biological research tools, services, and standards. In the following webinar, Dr. DeMars will discuss the need for standards in clinical laboratory testing and will expand on how ATCC supports this with authenticated biological and molecular reference materials. If you have any questions for Dr. DeMars, please use the chat function available through the webinar program. Questions will be answered as time allows at the end of the presentation, and any remaining questions, as well as the recorded webinar presentation, will be archived on the ATCC website at www.atcc.org.
Okay, let me start out with a brief introduction on ATCC. Founded in 1925, ATCC is a nonprofit organization with headquarters in Manassas, Virginia, and an R&D and Services Center in Gaithersburg, Maryland. ATCC houses a premier biological materials resource center and standards development and is a standards development organization. We have over 4,000 cell lines, over 70,000 microbes, and we also have genomic and synthetic nucleic acids as well as media and reagents. ATCC collaborates with and supports the scientific community with industry standard and innovative biological solutions. We have a growing portfolio of products and services with sales and distribution in 150 countries and 12 international distributors. As a not-for-profit organization, we adhere to admission, which is to acquire, authenticate, preserve, develop, and standardize, as well as distribute biological materials and information for the advancement and application of scientific knowledge. As a comprehensive standards organization, ATCC is committed to providing standard solutions in three key areas: products, standards, and services. ATCC's products are fully authenticated, characterized, and purity tested, having been accessioned for over 90 years, can offer breadth and depth of cell lines and microbial strains, as well as derivatives from those materials. Many of our products are recognized standards, recognized and available globally, collaborating with international government regulatory organizations. And additionally, ATCC has recognized standards development organization, which develops consensus standards, protocols, procedures developed to standardize practices in the life sciences to help ensure quality and reproducibility. The same rigor for product and standard development is incorporated into our services as well, where we provide customers with specialized needs to produce our or their proprietary material for research use only, CGMP, or under ISO requirements. Our current certification includes ISO 9001 for overall certification for quality management system, ISO 13485 for the certification for medical devices, ISO Guide 34 for the production of certified reference material, and ISO 17025 accreditation for testing, such as our cell line authentication services. In the case of standards for life science research published in December 2013 by Global Biological Standards Institute, or GBSI, they did a great job of summarizing the implication and importance of standards to the broader life sciences stakeholders. So you can see that in terms of looking at the stakeholders for the life sciences, there are academic labs and institutions where you need to have standards to ensure best practices are incorporated into the research process, to be able to produce more reproducible results, to decrease misinformation, protect reputation, enhance collaboration with industry. For the industry and investors, the need for standards is to reduce target validation and development failures, to improve translatability and commercialization of discoveries, enhance collaboration with academia, improve return on investment, and optimize the use of funds. On the government and nonprofit side, facilitate grant review process, optimize use of financial resources, protect reputation, improve public perception of life science research. And for journals and professional societies, standards are necessary to facilitate peer review process, protect reputation, decrease misinformation, and improve the public perception of life science research. So an important category of standards used in life sciences or clinical diagnostics is reference material. So what is reference material? Reference material is a material or substance, one or more of whose property values are sufficiently homogeneous and well established to be used for the calibration of a measuring system, the assessment of a measurement procedure, or for aligning values to materials. There are a variety of reference materials, including certified reference materials, standard reference materials, calibrators, and characterized genomic nucleic acids. Reference materials can be qualitative, either the presence or absence of a marker, for example, or quantitative, looking at the level of the expression of a marker. It should be obvious and intuitive that standards such as reference materials can help reduce unintentional differences between laboratories. And this is true for reagent consistency, consistency of laboratory processes, experimental design and analysis, and data reporting and sharing. Irreproducibility is a serious problem which not only can affect patients, but also impacts reputation, public opinion of research, and the use of time and resources. With regard to the importance of using reference materials, there is a recognized need and in many cases requirement for the use of established, fully characterized, globally accepted reference materials throughout the test development, validation, and quality management process. In the development of tests, there are often new technologies and new assays that require optimization and may need new reference materials. As the tests are validated to optimize sensitivity, specificity, robustness, and reproducibility, the standards may need to be altered or further developed, and many times with clinical diagnostics targeting mutations, the scope of variance must be captured by the reference material. And then for the ongoing quality of a launched diagnostic test and proficiency testing applications required by regulatory authorities, such as the FDA and CLIA, reference materials are necessary to ensure the ongoing quality performance of that test. The sample or reference material characterization that is required often increases throughout the test development process. Although there are many clinical diagnostic tests using various technologies and platforms, there are several categories of biomaterials that are consistently and effectively used as controls or reference materials. And those categories are synthetic oligonucleotides, which are easy to design and synthesize as the benefit, but the disadvantage is that they do not resemble complexity of the whole genome. The reference material of whole cells and genomic DNA, the benefit of those are that they mimic the complexity of the whole genome, with the disadvantage being that the genetic stability, rare mutations are difficult to obtain. Patient samples are a third group of reference materials, and the benefit is that they're extremely representative of the patient themselves, but the disadvantage is that they are not a sustainable resource. The properties of these materials that are attractive and that are considered in their selection for use include that they are fully authenticated, they have characterized genetic alterations, they have stable molecular profiles, they avoid contamination or misidentification, and they provide reproducible results. I wanted to take a moment to discuss a particular type of reference material highlighted in the previous slide and talk more about the potential variations of that material and the application and trade-offs to consider when developing appropriate standards. Genomic DNA is currently and often used by researchers and testing laboratories to provide positive and negative controls, particularly for tests that use patient DNA. In many cases, this material is easily obtained and easy to make, but used without extensive characterization. Depending on the test being used, it might be necessary to make sure that the control has a particular biomarker or even quantify that biomarker for a quantitative test. As that test is being validated and for QC and regulatory purposes, each lot of that material produced needs to be characterized and meet specifications and eventually have a certificate of analysis supplied providing measurement results with associated uncertainties. The increased characterization and measurement of these standards ensures high confidence in test results and are of high value, but are difficult to develop and take investment of time and resources and are an important element of approval or accreditation of that test. I mentioned earlier in the talk the importance of the global nature of standards and wanted to take a moment to provide a little bit more detail on ATCC as a global standards provider. Over 475 ATCC products are recognized by more than 30 government and commercial organizations. And those government and commercial organizations include over 10 international government organizations or agencies. ATCC has current and ongoing collaborations with U.S. regulatory agencies to develop reference materials for new diseases and applications and new technologies. And we have ongoing relationships and work in progress with the FDA, NIH, as well as NIST. Now I will discuss specific examples of standards provided by ATCC, both in cell biology as well as microbiology. For humans, a large focus of our cell line portfolio is cancer. We have hundreds of cancer cell lines derived from tissues and blood and feature a unique collection of normal and matched cell line pairs for melanoma as well as other cancer types, and have developed the first comprehensive catalog of somatic mutations from an individual cancer. We also have cell line genomic DNAs for the molecular diagnosis of cancer that are produced under ISO Guide 34 as certified reference material. We have tumor cell panels, which are annotated with gene mutation data and additional in-house testing, as well as isogenic cell lines, where we have created target variants of cell lines using CRISPR gene editing. These cell lines or isogenic cell lines are intensively validated on the genome, transcript, as well as protein level. We see here at ATCC that CRISPR gene editing as a technology with incredible promise for standards applications and is an active piece of our R&D program with our products and services groups. I wanted to highlight one recent foray into the cancer cell line-based standards. In July 2016, NCI selected ATCC to distribute cutting-edge cancer models to support the Human Cancer Model Initiative. HCMI is an international collaboration between the NCI, Cancer Institute UK, Wellcome Trust Sanger Institute, and the Foundation Hubrecht Organoid Technology. The goal of this initiative is to develop approximately 1,000 cancer cell models that better represent the hallmarks and diversity of human cancer and become standardized cell-based models. ATCC will develop the infrastructure to support the intake, authentication, production, quality control, marketing, and distribution of these cell lines. Variety of cancer cell types, organoid types, including rare and pediatric cancers, are included. Complete genetic analysis and de-identified clinical information about the patient tumors, including their response to treatment, will be included. So no presentation about standards derived from cell lines is complete without emphasizing the need for proper authentication. The cell lines ATCC provides are fully authenticated, performing CO1 and STR testing on our cell lines to avoid interspecies and interspecies contamination or misidentification. In addition to the authentication, ATCC cell lines undergo extensive characterization of tumor genetic alterations. And we are able to ensure stable molecular profiles, and we also can use these cell lines and controls for specific cancer marker tests. These cell lines can also be used as controls for specific cancer marker tests, especially in the area of FFPE, formalin-fixed paraffin-embedded samples, so that these samples are used for the testing of tissues to look at immunofluorescence, immunohistochemistry, or presence or absence of specific nucleic acids. In all these examples, controls from cell lines offer a more replenishable, quality-controlled option over human material. Moving on to ACC standards and reference materials for infectious disease, we offer highly characterized microbial strains for assay development, genomic nucleic acids, quantitative standards, DNA from bacteria, fungi, protists, and viral strains, RNA from viral strains, synthetic nucleic acids, genetic surrogates, and wanting to cite a specific example with Zika, we have accessioned several recent strains, have genomic and DNA isolated and synthetic RNA made, as well as inactivated strains. In terms of authentication on the microbiology side, ATCC utilizes both classical and modern techniques. We perform phenotypic analysis looking at colony morphology, cell attributes, and biochemical analyses. Look at genotypic analysis with sequencing, toxinotyping, and ribotyping. We conduct proteotypic analysis using the VITEC mass spec. And we also do functional analysis looking at serotype, drug resistance, and virulence. ATCC also produces a category of highly characterized products referred to as certified reference material. These materials are used to challenge assay performance, validate or compare test methods, establish sensitivity, linearity, and specificity during assay validation or implementation, benchmark critical assay performance during development or validation for regulatory submissions and production lot release, and then also for testing and calibration on ISO 17025 accredited laboratories. All certified reference materials from ATCC are accompanied by a certificate of analysis. ATCC is also a provider of material for proficiency testing programs. We have dedicated, highly characterized stocks that are used in proficiency testing panels that are consistent, safe, reliable, and traceable to their source. They are test-relevant, actionable targets, representatives of contemporary strains, genetic variations and mutations, new emerging strain variants, new technology developments, and next-gen sequencing, MALDI-TOF, and multiplex technologies. One way to ensure quality and consistency is reflected in the way that we approach production of our material, with the net result that the preserved cultures remain as close as possible to the original culture. So we have a process by which we start with the culture. Seed stock is developed and archived for future replenishment. The distribution stocks then are generated from a seed stock and are used for distribution. Authentication compares the seed distribution and initial cultures, and working cell banks are established and are essential for reproducibility and minimizing passage. Up until this point, we've been talking about the physical and material standards which ATCC provides, and now I would like to switch to the focus to consensus standards. ATCC has a standards development organization which is accredited by the American National Standards Institute, or ANSI. We convene and manage an international consensus group to produce written standards. We develop best practice standards in life sciences laboratory testing and promote their use globally using a consensus-driven process that balances the viewpoints of industry, government, academic, and clinical professions. And it ensures the reliability and reproducibility of biological materials, establishing institutional and lab systems that ensure research quality. Examples of the ATCC SDO consensus standards include ASN 0001, which is the standardization of in vitro assays to determine anthrax toxin activities. 2 is ASN 0002, authentication of human cell lines, standardization of STR profiling. And the third one, which is ASN 0003, which is species-level identification of animal cells through mitochondrial cytochrome C oxidase subunit 1, CO1 DNA barcodes. So in summary, I hope that this has provided an overview on the importance of standards in clinical diagnostics and the efforts of ATCC to develop and provide valuable quality standards. ATCC is an innovative global partner for high-quality, authentic biomaterials, standards, and services. ATCC collaborates with industry and academia to support scientific research and breakthroughs through the continual development of new standards and reference materials. And we have scientific expertise internally as well as through extended global partners to reach more than 150 countries.
Thank you, Dr. DeMars, for the excellent presentation, and thank you everyone for attending the webinar. Please visit us online at www.atcc.org/webinars to register for additional ATCC webinars. For more information about ATCC's standards program, please visit us online at www.atcc.org/standards. Thank you again and have a great day.